Hello, everyone, and welcome to the Science and Chill podcast. I'm your host, Brady Holmer. My guest today is Dr. Charles Brenner. Dr. Brenner is the Alfred E. Mann Family Foundation Chair in Diabetes and Cancer Metabolism at City of Hope National Medical Center. In 2004, Brenner discovered nicotinamide riboside, NR, as a vital precursor of nicotinamide adenine dinucleotide, NAD, which is made available by nicotinamide riboside kinases, or Nrks, that are conserved between yeast and humans. In 2007, Dr. Brenner's lab discovered a second pathway by which NR is converted to NAD and showed that NR can extend the lifespan of yeast cells by elevating NAD levels and increasing the activity of NAD-dependent SIR2 enzymes. In the past decade, Dr. Brenner has made multiple contributions to NAD metabolism, which include engineering a yeast strain to convert inexpensive NAD precursor vitamins into NR, solving the crystal structure of human Nrk1, developing the methods for quantitative NAD metabolomics, and demonstrating the activity of oral NR in human models of fatty liver disease, obesity, type 2 diabetes, diabetic and chemotherapeutic neuropathy, heart failure, and central brain injury. Dr. Brenner is also leading the first clinical trial of NR, which established safe oral availability in humans, and he is currently focused on the maternal and neonatal effects of oral NR and translating animal discoveries into evidence-based, safe human clinical practice for conditions of metabolic stress. Now, Dr. Brenner is clearly a well-credentialed scientist, given the extensive background I just described, but my main impetus for inviting him to come on the podcast today was his recent and public criticisms of the longevity field. In particular, Dr. Brenner has several recent posts in which he has directly called out prominent longevity scientist Dr. David Sinclair for what Dr. Brenner describes as spreading misinformation regarding what we know about aging and whether or not we can slow or reverse aging. We spend the last 20 to 30 minutes or so of this podcast discussing his critiques and where he believes scientists in the longevity field may be overstepping. Other topics that we get into in the podcast include basics on human metabolism, nicotinamide adenine mononucleotide, and nicotinamide adenine dinucleotide, or NAD, and the importance of the NAD system in physiology, what types of things perturb the NAD system, and how this may contribute to diseases, aging, and ways in which we may be able to boost the NAD system through diet and supplementation. I think a lot of these interventions you will find interesting, as well as a lot of insights into what things can cause the NAD system to be disrupted. I will say that at times we do get pretty technical in this podcast. Dr. Brenner gets into some of the more complex biochemistry surrounding metabolism and the NAD system. But I do really encourage you to listen to the entire interview and everything we chat about, because Dr. Brenner is such an eloquent speaker on a lot of these topics. And I think that even if some concepts are a bit higher level, you'll learn a lot about biology and physiology through what he has to say. I surely did. This was almost a little bit of a crash course in biochemistry for me. If you enjoy this podcast and would like to support it, there are several ways that you can do so. The first is by becoming a supporter on Patreon. With Patreon, you can support the podcast with a monthly donation on any level you'd like. You can start showing your support right now by going to patreon.com/Brady Holmer. You can also just check the show notes for a link to my Patreon page. Thank you so much to all of my current supporters on Patreon and to those who will be supporters in the future. You can also make a one-time donation to help support the podcast through PayPal or Venmo. All of these support options can be found on my website by going to bradyholmer.com/donate. And there's also a link in the show notes to that support page as well. And finally, checking out podcast sponsors is also a fantastic way to show your support and try some new products. There are two partners for today's podcast. Both of these are products that I use frequently and absolutely love. The first is Element Electrolyte Drink Mix. Element is a sugar-free, carbohydrate-free, tasty electrolyte drink mix containing scientifically based formulas and ratios of sodium, potassium, and magnesium. The single serve packets are great to add to water for any time surrounding a workout or just throughout the day. I'm routinely using about 3 packets of Element per day in my water, and they come in a variety of awesome flavors, including citrus salt, which is actually my favorite flavor, raspberry, and watermelon. They also have a bunch of different flavors, including some spicy flavors that you can find on their website. And if you'd like to sample Element, you can try a free 8-pack sampler, deal specific for my audience, by going to drinkelement.com/science-and-chill. There's also, again, a link in the show notes to that special offer. I have also partnered recently with Health via Modern Nutrition, or HVMN, to bring a special offer for my audience on their brand new Ketone IQ exogenous ketone drink. Now, ketones are sometimes called the fourth fuel and can be used by our muscles, our heart, and our brain for energy. With exogenous ketone drinks like Ketone IQ, you can rapidly elevate your blood ketones and put your body into a state of ketosis. Now, this is something I have actually confirmed with self-testing, results of which I have published on my blog and on Twitter, where these products actually do elevate your ketones within about 60 minutes of drinking them. So I can confirm they do put you into ketosis, and I also can confirm that they make you feel awesome. And studies, again, plus my personal experience, have found that ketones can help with things like mental performance and mental clarity, athletic performance, and just maintaining energy levels throughout the day. So if you've not yet tried ketones, and there are many available products out there, I really encourage you to give Ketone IQ a try. This is kind of the newest iteration of exogenous ketones out on the market. And for a limited time, you can get 10% off of your order of Ketone IQ by going to their website, using the link in the show notes, and entering the code Brady10 at checkout. Again, if you use the link in the show notes to learn more about Ketone IQ and enter the code Brady10 at checkout, you can get 10% off your order of Ketone IQ. This is a product that I know you're going to enjoy. And last but not least, if you enjoy my podcast, then you may also be interested and enjoy some of the writing that I'm publishing on a weekly basis, which also includes my weekly email newsletter that I send out every Friday called Physiology Friday. If you'd like to check out my writing and subscribe to receive my weekly posts and my email newsletter, please visit bradyholmer.substack.com. You can also find a link to that in the show notes. And if you subscribe, I hope that you enjoy. Without further ado, please enjoy my conversation with Dr. Charles Brenner. All right. Dr. Brenner, welcome to the Science and Chill podcast. Thanks so much for joining me today. I'm very excited to chat about many topics that we have planned.
Great. Okay. I'm looking forward to it.
For sure. So before we begin, could you please just give listeners a brief background into your scientific training, maybe a little bit of a bio on who you are and kind of where you're currently working and what you are currently working on?
Sure. Okay. Let's see. I'm currently the Alfred E. Mann Family Foundation Chair and Professor of Diabetes and Cancer Metabolism at City of Hope National Medical Center outside of Los Angeles. Prior to that, I spent, I think it was 11 years as the head of the biochemistry department at University of Iowa. Prior to that, I spent 6 years on the faculty of Dartmouth Medical School. That's where I discovered the vitamin activity of nicotinamide riboside kinase pathway in NAD metabolism. I've been doing NAD for 20 years or so. Actually, before Dartmouth, I was on the faculty for 7 years at Thomas Jefferson University. I was a postdoc at Brandeis, 93 to 96, where I did X-ray crystallography. My PhD is in cancer biology. I was earned in the biochemistry department at Stanford. So I'm originally from the East Coast. I'm a Bostonian. I went to college a gazillion years ago, graduated in 1983 from Wesleyan University, spent 5 years in biotech, in the San Francisco Bay Area, then 4.5 years at Stanford getting my PhD, basically in the Stanford biochemistry department. And postdoc, as I said, at Brandeis, been a faculty member since 1996. This is my 4th institution, probably my last one. I'm 60, but I've moved around a lot. I've really enjoyed my time in science. And essentially, my lab does nicotinamide, adenine, dinucleotide metabolism all day, all the time. We are still finding additional genes, additional regulatory pathways. These are the crown jewels of metabolism, NAD coenzymes, yet they're not locked inside of a safe in a vault patrolled by armed guards. They're actually exposed to the elements of metabolic stress. We find conditions in which the NAD coenzymes are disturbed. A lot of times nicotinamide riboside kinase genes go up, and that gives us indications that something like nicotinamide riboside may be protective. In other cases, we find an NAD system that's dysregulated, that we can kill those cells like cancer cells that have dysregulated NAD. And so that's what we do.
Awesome. So, and we're certainly going to get into maybe a lot of the nuances regarding nicotinamide riboside, NAD metabolism. I think we should maybe go back a little bit though for listeners and just give everybody sort of maybe a little biochemistry metabolism 101 kind of.
Sure.
We're going to be discussing a lot of topics today regarding metabolism and when we get into aging and longevity. So I would like to sort of just set the stage and maybe talk about some terms. So let's just begin kind of at the basics and just what is metabolism? We hear this word thrown around a lot, like, oh, you need to boost your metabolism, or metabolism slows with aging. So what in general kind of is metabolism? Can you give us a definition of that?
Yeah, the way I like to define it is metabolism is the set of reactions that converts everything that we eat into everything that we are and do, and further allows us to repair ourselves. So the first conception of metabolism is fuel to ATP metabolism, oxidative metabolism, where you take in protein, fat, and carbohydrate, you quote, burn it, you use oxygen to oxidize stuff. And if you fully oxidize foodstuffs, then what you release is CO2, carbon dioxide, and the yield is kind of ATP, biological energy that is used to run things, to flex muscles and circulate things and catalyze difficult chemical reactions. That's kind of the first thing that we teach in metabolism, is kind of glycolysis, like sugar oxidation, amino acid oxidation. That's kind of hard, right? Mm-hmm. And then fatty acid oxidation, which is very straightforward, where everything ends up making acetyl-CoA and going into TCA cycle. That's not all of metabolism, because you have to make stuff, right? Everything in your body, unless you have an artificial hip or artificial lens in your eye or something like that, you made yourself. And so really what happens is that we eat food. Some of the food is totally broken down and sort of fully combusted to generate ATP, but a lot of it just goes to intermediates where you remake stuff. And that's where the fun is, really, of biology, is making and repairing stuff. So metabolism is and crucially, electron transfers, harvesting the energy of a high energy electron on a molecule called NAD, nicotinamide adenine dinucleotide, is critical, not only for fuel oxidation, where the electrons end up on NADH, and NADH starts the electron transfers to generate ATP, but those same electrons can be put onto NADPH to make stuff. So electron flow is really critical to metabolism. The energy for everything, if we had enough time, we could trace back to the sun and photons getting excited that end up basically growing the plants that we either ate, or the animals that we ate the plants that came from solar energy. But basically, all metabolism is everything you eat into everything that you are, and high energy electron transfers are incredibly important, and they're mediated by 4 NAD coenzymes.
That was a beautiful description of metabolism, probably a textbook description of metabolism. So now you mentioned these NAD coenzymes, NAD, NADPH. So now can we get into sort of what these coenzymes are, why they're important in metabolism, and then sort of, their importance in physiology? You just spoke about how obviously we need these to create energy, and essentially, the way in which we create energy and build up things is essentially through electron transfer. We have this electron transport chain, which is just kind of like a big battery, basically, within all of our cells. So can you talk a little bit about NAD metabolism, why that's so important for physiology?
Sure. Well, there's not an electron transfer in every single biochemical step, but there are electron transfers in all major pathways. And you can't get the reactions to go unless you transfer electrons. And the other thing about metabolism is that these reactions are not lossless, right? So, and the losses of energy get effectively wasted as thermal energy, right? And so if you were to calculate the amount of heat that you could generate from burning, glucose, and you could calculate the difference in kind of driving potential between ADP and ATP, biology is very efficient at generating ATP and not generating a lot of heat. There's other conditions in which you sort of want to generate heat, and biology can do that too. But basically, these electron flows are essential for fuel oxidation, as well as making all of the things that we have to make. And while NAD and NADH, NADP, NADPH dependent steps are not present at every single step, they're present in essentially all pathways. And they preserve the high energy state of electrons that come from food. There are lower things in the electron chain that can accept lower energy electrons. Ultimately, oxygen is the energy acceptor in fuel oxidation, and H2O is generated, but that's a very low energy electron. The electrons that are on NADH are at a much higher energy level, and they can do more things. They're sort of like, if you imagine a waterfall or a water mill. If any of you have been to New England and have seen a good old-fashioned water mill, the high energy electrons basically have further that they can drop, and they can do more work. And the electrons that are on NADH and NADPH can do a lot of biological work. That's why they're so important.
And so is that just due to the amount of the driving potential or that those electrons have, or why are they able to, I guess, do more work?
They are literally, in the electron transfer chain, you see, so now you're getting into biochemistry nerds are following us. So we'll try not to, I mean, I don't know who listens to your podcast, but
it'll be a diverse kind of in terms of the backgrounds of biochemistry. So I'll be sure to include a picture of the electron transport chain in the show notes, at least so people can maybe follow along.
NADH is a higher energy electron donor than coenzyme Q. Coenzyme Q and FADH are kind of intermediate. Cytochrome oxidase is much lower, and oxygen is the ultimate electron acceptor, is the lowest. So it is the energy potential of the electrons as they flow through the chain.
And so obviously this system is so important, not only important, I guess, but vital, necessary for life. So what perturbs the system? I know your lab studies now NAD metabolism, kind of what goes wrong. So obviously we're interested in, when it works right, but oftentimes in physiology and disease, things go wrong. So what perturbs our NAD systems in terms of environmental stresses, things like that?
Well, that's a great question. So I'll start with 2 or 3 things that were known before our work. And that's ethanol, reactive oxygen stress, and DNA damage. And I'll tell you how those 3 things disturb the NAD system. And then I'll name 5 or 6 more that we and others have much more recently discovered.
You're not going to tell us alcohol's bad for us, are you, Dr. Brenner?
So alcohol, in alcohol metabolism, which largely occurs in the liver, alcohol is converted to acetaldehyde and then to acetate in 2 NAD-dependent steps. And so in so doing, the liver becomes more reduced. So that this is called redox biochemistry. NAD plus is the electron acceptor. NADH has received 2 electrons from something. It could have received them from ethanol or from acetaldehyde, and that generates NADH. And so NADH is good stuff. I mean, at the intermitochondrial membrane, NADH provides the electrons that initiate the electron transfer chain at complex 1. But if you flood hepatocytes with ethanol, you put the fuel oxidation system in a reduced state in which they can't really burn fuel because they're so reduced. They're kind of constipated with electrons. And so that's a system that disturbs the NAD system. That was known kind of classically. Reactive oxygen stress also disturbs the NAD system by kind of churning NADPH. NADPH provides the electrons for glutathione reactivation. And so that was kind of classically known. And then kind of intermediate between classically known and really current is DNA damage, because when DNA is damaged, an enzyme called PARP1 and PARP2 as well get excited and basically use NAD as a consumed substrate. So not as a source of redox biochemistry, but as a source of what's called ADP ribose. So if we were to draw the chemical structure, and maybe you can put it on a web supplement, the ADP ribose moiety piece of NAD gets polymerized in order to basically signal repair enzymes that there's a DNA damage event that has to be taken care of. So that takes down the levels of the central catalyst of metabolism in the case of DNA damage. Now, we further have found many other conditions of metabolic stress that disturb the NAD system. So Anthony Salve and others found that noise sufficient to produce hearing loss, takes down NAD in the cochlea. Also, other folks have shown that high noise induces expression of the nicotinamide riboside kinase genes. So NAD is under attack, is the way we term it. And the nicotinamide riboside kinase genes get ramped up, which basically says that in that condition and other conditions I'll tell you about, the damaged cells are looking for NR, nicotinamide riboside, to replete, to rebuild their NAD supply. Why NR? Well, when Matthias Nrks and I looked at a mouse model of heart failure, was the serum response factor, the cardiac knockout of the serum response factor mouse, which gets a dilated cardiomyopathy, develops into full-blown heart failure. The nicotinamide riboside kinase 2 gene is the most highly induced gene in the failing heart. Failing heart is not making a lot of ATP, so it has low levels of phosphocreatine, it has high levels of AMP. This activates AMP kinase, right? Low energy signal. And it turns out the nicotinamide riboside kinase 2 gene promoter gets turned on by AMP kinase. Now, why? What's the logic of this? The logic of this is that when then the heart's supposed to be making ATP, right? It's supposed to be, that's classic fuel oxidation energy production, right? Fatty acids or glucose. In that low energy crisis where AMP kinase is activated, the signal pathway ramps up nicotinamide riboside kinase expression because it takes cost cells less ATP to rebuild their NAD from NR than from any other source. Nicotinamide riboside basically already has the ribose on it, so it doesn't take ATP dependent processes to generate the PRPP for nucleotide synthesis. If the failing cardiomyocyte can find some NR, it can rebuild its NAD supply without taxing its ATP. So it's very interesting. Now,
so is it sort of just looking for like an alternative pathway to like produce energy then, since it's, energy production is disrupted and it's not able to produce like ATP? Is it sort of like, is this finding a different pathway by upregulating the NR kinase?
Around NAD. There's no way around, as it needs for those cells to survive and not go into, there's a number of different types of death pathways that get invoked: apoptosis, pyroptosis, necroptosis, that can interface with attacks of NAD. They need NAD, but those cells don't want to commit a lot of ATP, which is also under attack, in order to rebuild their NAD supply.
I see.
So if they can find NR, if there's any NR available, that's going to be the preferred NAD precursor. So eukaryotic cells in toto have 4 ways of making NAD. They can make NAD from what's called a de novo pathway, which starts all the way from tryptophan, and it goes through like 8 biochemical steps, 8 enzymatic steps to convert the amino acid tryptophan into NAD. Is very inefficient. Tryptophan is used for lots of different things, including making protein, right? Serotonin, kynurin metabolites, all kinds of things. So you have 6 enzymatic steps before you even have something that resembles NAD. You would have a hard time, seeing the carbons and nitrogens in tryptophan that end up contiguous in a fully formed NAD. So that's a difficult and inefficient process. And then there's 3 what we call NAD precursor vitamins: nicotinic acid, niacin, nicotinamide, and then the one that was not thought to be an NAD precursor until we came along in 2004 and showed that it is NR, nicotinamide riboside. Those 3 vitamin precursors of NAD have 3 different gene/enzyme encoded pathways to make NAD from those precursors. And it costs less ATP to make NAD from NR. So that's the kind of use case for NR. Now, so that's great for the mouse with heart failure. If it happens to be in, a cage that's being fed with NR, then it does way better than those that are not so supplemented. But, this AMP kinase activated nicotinamide riboside kinase gene transcription system, this has been around for, millions of years, right? This gene regulatory pathway doesn't anticipate the discovery of NR as a vitamin, in 2004, and somebody in Paris feeding it with NR. That pathway exists for a reason, which suggests to us that even though it's difficult to detect circulating nicotinamide riboside, there is endogenous nicotinamide riboside in mice and in people. And one of the things that we're working on in our lab is what are the donor cells and what are the recipient cells for distribution of nicotinamide riboside, because cells don't make nucleosides intentionally. Cells make nucleotides. But if you go to your biochemistry textbooks and you look at how nucleotides are made, cells make nucleotides and then they salvage either the bases or the nucleosides. So it suggests to us that NR in the failing heart is actually coming from other cells, if it's not being fed to the animals.
Right, and I guess that was going to be one of my questions, whether we have endogenous precursors for NAD, or whether we need to say consume them, since you mentioned that they are kind of like vitamins.
Right.
Are they essential or non-essential?
Yeah, and the answer is conditionally essential. So there's sort of low level essentiality of NAD precursors that was defined a long time ago, actually. So 100 years ago in the American South, there was a rampant pandemic called pellagra, a disease that you don't see any longer. The National Health Service, which was the precursor of the National Institutes of Health, in fact, today it's still administratively bigger than the NIH, funded the first public health service awardee, Dr. Goldberger, to go down to the American South to kind of figure out pellagra. And everybody told him he was going to find an infectious disease because it was rampant in places like hospitals and jails. And when he went to the hospitals and jails, he found that the imprisoned and the patient populations had pellagra, and the jailers and the staff in the hospitals didn't have pellagra. So it didn't make sense as an infectious disease.
And is pellagra, is it related to like a vitamin B deficiency or something like that? Or do I have that incorrect?
No, so Goldberger said, I think it's the food. And he gave them fresh food, and he could reverse pellagra, and he could show that, I mean, it was just beautiful public health work. And he could show that fresh food was pellagra preventative, had a PP factor, pellagra preventative factor, and also could treat. And he didn't know, what in food, like, for example, milk, was one of the things, eggs, meat, lots of things would prevent pellagra. And, he died without knowing what in food, prevented pellagra. Then in the late 1930s, we get the elucidation of the chemical structure of NAD. At the time, it was called DPN, diphosphopiridine nucleotide. And Otto Warburg, who basically escaped from Nazi Germany and was one of the leading biochemists of his time, elucidated the chemical structure of NAD in essentially the same year. Conrad Elvehjem and colleagues in Madison, Wisconsin, found that nicotinic acid and nicotinamide would prevent a pellagra-like condition in dogs. We won't get off on a tangent on how they got those dogs and how they made those dogs sick, because it's kind of an unpleasant and doesn't pass research ethics of 2022. But basically, Elvehjem finds that vitamins that are pellagra preventative, that have a nicotinamide or nicotinic acid base, diphosphopiridine nucleotide, NAD, DPN, as they called it in that era, has a pyridine, nicotinamide base. So essentially, in 1938, we start to realize that basically the pyridine unit of NAD is can be limiting in the human diet. And then in the 40s, the tryptophan as an NAD precursor was worked out. And then in full circle, in order to prevent pellagra on any kind of horrific diet, the corn rations and lard was kind of the most pellagrogenic diet, in the American South. And by the way, the corn was milled in a steel machine that was being, widely distributed through the South, which was a different method than corn grinding in Mesoamerica, where people have been successfully on corn-based diets for millennia. But it turns out what's called the nixtamization process of grinding corn with stone leaves a lime alkali residue, in the corn meal, in the masa. And that alkali basically releases the NAD precursors, such that in Mesoamerica, the Native Americans ate a corn-based diet and never got pellagra. They
That's
figured
so,
that out a long time ago.
yeah, that's so
And
fascinating because I mean, to me, you just think about all, the molecular techniques and things like that we've been studying, but these like ancient civilizations, they had none of the techniques that we have available, but somehow we're able to just like intuitively, figure out that this process of, grinding up the corn eventually would allow them to eat like this corn-based diet. It's just amazing how
I don't think
at
it's
least I find it
intuitive. I
amazing how
think
it
it's
happens.
selection, man. It's
Hmm.
that, when, well, they're growing corn. They found the best rocks to, to pound it, the best tools to pound it. And actually, here in Los Angeles, every year there, I think it's around this time of year when there's, March Madness is a big basketball tournament. There's a tortilla tournament in Los Angeles that is run by a columnist named Gustavo Arellano, whom I follow on Twitter. And he discovers all these amazing stories in the tortillerias that he visits in order to do the tortilla, matchups. And he interviewed somebody that still sources the limestone from some quarry in Mexico in order to get the right consistency of the masa. And of course it has, the alkali that is needed to make it a good source of B3. But basically, in the larger question, where do NAD precursors come from? And the answer is, we eat cellular stuff, right? We're eating plants, animals, and fungi. NAD coenzymes are the central catalysts in all cells, whether they are plants, animals, or fungi. And so there are NAD coenzymes in all of the things that you're eating. Back in the old days of, ice bucket biochemistry, if you wanted to, purify NAD-dependent enzymes or NAD-dependent NAD itself, you could get a piece of beef liver, or you could go to the bakery and get a big brick of yeast, and you could, grind the yeast paste, or you could homogenize the liver. And obviously, if you were to cook meat, or you were to cook vegetables and minimally process them, you would have lots of NAD coenzymes in that mix. Now, the more that you process the food, the more you're breaking it down. So flour that is highly processed or is ground with metal, in the last many decades in the United States, has actually been enriched with niacin.
Mm-hmm.
Okay, you'll see niacin enriched, so that even on a diet where you go through the grocery store and you don't eat any of the fresh foods that are in the perimeter, you eat only in the aisles where the crackers are sold, you probably wouldn't be getting full-blown pellagra from those processed foods. But you asked me, what are the diseasing conditions of metabolic stress in which the NAD system is under attack? I told you classically alcoholism and reactive oxygen stress, and then also DNA damage. But so sun damage is going to break DNA. But we've also, and I think I told you noise-induced hearing loss, but we've also shown that overnutrition in a mouse model, if you feed a mouse a high-fat diet, their liver NAD system comes under attack. They have low levels of NAD metabolites, particular NADP and NADPH. Probably time zone disruption. There's not a lot of that right now during the pandemic, but shift workers or people that travel a lot of time zones are disrupting their circadian system. Circadian system, regulates NAD synthesis. Inflammation and infection. We recently showed that coronavirus infection in model systems, including human corpses, disturbs the NAD system by activating transcription of a whole bunch of enzymes in the PARP superfamily. They degrade NAD in an attempt to fight the virus. Lots of conditions of metabolic stress disturb the NAD system, and potentially aging as well, such that one-a-day type vitamins have always had levels of B3, either nicotinic acid or nicotinamide in them. And based upon our work, there's been commercialization of nicotinamide riboside. Full disclosure, I'm the chief scientific advisor of Chromadex, which developed True Niagen as a new dietary ingredient and as an FDA kind of recognized, generally recognized as safe over-the-counter supplement. It's not intended for any disease or condition, but from the model system work, you would expect that disease and conditions of metabolic stress that disturb the NAD system would be those in which NR, potentially has the most benefit. So in the laboratory, as well as in clinical studies that are around the world, like 50 of them, NR is being tested for its activity against things like fatty liver, diabetic neuropathy, chemotherapeutic neuropathy, cognitive impairment. There was a positive study done, very small, on Parkinson's disease, in which NR had some activity in combination with 3 other supplements. NR accelerated time to recovery from COVID, which we're very pleased that was kind of based upon our preclinical work with NR. So that's kind of the use case for NR.
Yeah, I think that the use cases for disease certainly make sense given the wide range. It almost seems like in most diseases, NAD metabolism is going to be disrupted in some way. But then it also kind of just makes sense that in general, people, even quote unquote healthy people, could benefit from either a B vitamin supplementation on the regular or something like NR supplementation. Because from what I hear from you, a lot of things, almost everything just living disrupts the NAD system. I mean, we're constantly under a barrage of like DNA damaging environmental stressors, whether it's noise, whether it's radiation, if we're flying a lot, changing time zones, even just the sun. So I mean, our cells are undergoing DNA damage all of the time, essentially. And so, that could just at least suggest that, supplementing with B vitamins or NR is going to be helpful in general, because the NAD system is always being disrupted in some sort of way, correct? I mean,
it's a primary, target of inflammation, right? So we've shown that the pathway by which coronavirus turns on these PARP enzymes that attack cellular NAD is common to all things that induce interferons, and that's a lot of things. So that's it's almost anything that activates the innate immune response. So double-stranded RNA is like dozens of viruses, bacteria that make lipopolysaccharide. I don't know how it works, but dust mites, which is an animal irritant, activates the innate immune system. And so chronic inflammation is obviously very common and maybe a kind of tonic stimulation that challenges NAD. And then, like I said, tryptophan is not, you get tryptophan in your diet, you get NAD precursors from breaking down food in your diet. you could basically supplement with three things. Nicotinic acid, niacin, is considered a drug that over the years, some people have taken to control their cholesterol. So for some people, it increases their HDL, lowers their LDL. One of the problems is that there's flushing. So at hundreds of milligrams, which is when you're trying to really, so the amount of niacin or nicotinamide to prevent pellagra is like 15 milligrams a day. So very, very low. The amount to do something about cholesterol gets into like grams a day of niacin. But almost everyone has a flush reaction where your face turns bright red and it's like hot and uncomfortable. Nicotinamide and nicotinamide riboside don't produce a flush reaction. And the advantage in the use case of NR is that in many of these conditions of metabolic stress, the NR kinase pathway is upregulated. So that's why there's been so much interest in NR as a supplement that could keep NAD levels high and could make use of the NR kinase pathway being upregulated for the cells that kind of need it the most. But right now, the people that are taking NR are taking it because they perceive that they are, feeling better. There are, trainers in pro sports teams that are giving it to all of the players because they're involved in like a, weekly high stress, high, contact sport where recovery, and they may actually have data on athletic recovery from their athletes, but there's not a ton of those types of things that have been tried. But we know that NR is safe, and we know that clinical trials are being done for disease and conditions of metabolic stress. By the way, another one I'm really interested in the lab is postpartum. So postpartum, I like to describe as the mother of all metabolic stresses, because mom has just produced another, human being, or in the case of rats, maybe 10 of them. And her metabolism, changed very dramatically at, conception in order to produce an environment for, embryonic and fetal development. And then at parturition, there's another remarkable switch where macronutrients are being mobilized from her adipose and her liver to the mammary biosynthetic program to produce 100% of what is needed for the offspring until they're weaned. Now, whether a human mom chooses to do that 100% for, or supplement with formula or whatever, that's kind of her decision. But her biological program allows her to do that through an amazing switch that's mediated by prolactin and other hormones. And we found that a new mom's liver, I'm talking mouse and rat here, but I have some high confidence that this is true in humans as well, that a new rodent mother's liver is under significant NAD stress during this program. And that by supplementing the mom with nicotinamide riboside, she can produce more milk. She has an advantage in postpartum weight loss. And the milk seems to be much higher quality because the offspring brain development is advanced, and their physical capability and even anti-anxiety phenotypes and learning and memory are advantaged into the offspring adulthood, which is pretty remarkable. So
Oh,
that's
yeah.
another thing that we're really interested in.
Yeah, that's fascinating. And did you say that studies were being done by your group or others? And if they are being done, so would say this supplementation be something that starts immediately after they give birth, or maybe starting a little bit before they give birth?
Yeah.
What kind of
So,
protocol would that involve?
okay, so let's distinguish between rodents and people and
Sure.
pre, during pregnancy versus postpartum. So we have done an experiment where we supplemented with NR during pregnancy, and we're going to have something to say about that fairly soon. We've also compared different NAD precursors in the context of postpartum rats, and we're going to be presenting that probably in June at the NAD meeting, and that will be out in bioRxiv and be in review sort of late spring, I would say, 2022, June-ish. And then there's a group at UC Davis that is looking at moms of neonates that is doing a clinical trial of moms of neonates who are basically milk insufficient, and for whom the neonatal nutrition is really, really important. And so they're testing in women whether nicotinamide riboside will improve the quality and the quantity of milk production. And it'd be very, very interesting to look at, and I think the group up at UC Davis also wants to look at whether milk insufficient full-term moms will benefit from NR as well, but that's not been tested. But the idea is, from our initial publication, that NR could be a mom's vitamin. So there are kind of pregnancy vitamins, like folic acid, right, is considered a vitamin for mom during pregnancy, but there isn't a strongly developed concept of postpartum supplementation, and this is potentially, a rationalization for that, that based upon preclinical work that will be tested in women.
Yeah, sure. Yeah, you hear, a lot about prenatal vitamins, but not much about postnatal vitamins. So that'll be interesting data to see when that work eventually does come out. So thanks for kind of touching upon that. It's not something I plan to talk about, but definitely an interesting area. I would like to sort of not necessarily switch gears, but I do really want to talk about aging. So we talked about aging as one of the conditions which, well, supposedly, I guess you'll tell us a little bit about that, disturbs the NAD system. And you hear a lot in the aging literature that, NAD levels decline with age, and maybe this is one of the primary drivers of aging. So what does it mean, I guess, for someone to even say this, that with age, say, NAD levels go down? And then secondly, to build upon that, based on your work and that of others, can we confidently say that NAD levels go down with age? And then maybe to begin that, we don't have to go in depth on this, but maybe you could briefly just discuss how we even measure NAD levels in the first place, whether if we say they decline, is this tissue specific, or are we able to measure them in the blood and get an idea of NAD in the whole body?
Yeah, sounds good. Let me take 4 minutes to refresh water, coffee. Is that okay?
Oh, of course, no problem.
Okay.
I can just cut all that out. Yep.
Okay, sounds good.
All right, good to go.
So why don't I take the last question first, which is, how do you measure NAD?
Perfect.
So the gold standard way to measure what we call the NAD metabolome, it was developed by our group. It's quantitative targeted NAD metabolomics, and the method is LCMS. You need internal standards. And we developed it at University of Iowa, and it's been further refined at City of Hope, and it gives you the picomole amounts of NAD, NADP, and more than a dozen other NAD metabolites. It's actually kind of difficult to preserve NADH and NADPH for technical reasons, but you get an absolute amount of NAD. You can denominate that per milligram of tissue or per microliter of blood or something, so that you really know what the levels are. NAD is also reported by others from non-targeted metabolomics, where there are hundreds or thousands of metabolites and they're just looking at fold change. Like, they really don't know how much is there, but if they're looking at different conditions, they may be able to say there's more in this sample than in that sample. But the metabolites are not put on the same scale. Like someone could say there's a threefold increase in nicotinamide, but if nicotinamide levels are 1% of the level of NAD, then that threefold difference may not have a biological meaning. So it's a little bit obfuscating in some ways, or not as clear as people think it is. Then the worst measurements are the ones from what's called a cycling assay, which is the most common. And there are these, basically redox sensitive dyes, resazurin, MTT, and so forth, where there's a kit and it contains something like alcohol dehydrogenase and diaphorase and NADH standards, and those are not good ways of measuring NAD, and they certainly can't be used. It's like a PCR assay that tells you something that's there, but you can't use it as a way to quantify. And so some of the most headline-grabbing reports that NAD decline in aging were done with the worst tools. So it's widely believed and reported that NAD declines in aging. I know from really well done quantitative targeted metabolomics that NAD comes under attack in many conditions of metabolic stress, and I've seen recent data that indicate that in human beings with age, seems to be a depressed NAD system in muscle. There's lots and lots of reports of hepatic, liver NAD declining in rodent systems over time. I've not really seen convincing evidence that blood NAD declines in healthy people, but I have seen and measured myself blood NAD levels from people with mitochondrial myopathies, where they have a phenotype in their muscle and they present with a low blood NAD metabolome. So there are human beings that have, human NAD is, the human NAD systems in multiple tissues are almost certainly challenged by things that occur in age, whether they are directly a function of time or whether those challenges occurred over time episodically, like as a function of infection or sun exposure or something else, I don't really know. And I don't think anybody else knows. But I'm kind of the person in the field that when somebody says something, I ask them, how do they know that? And what do they mean by aging? And is it something that occurs over time, or is it time itself? And I think those are some of the unresolved questions.
Yeah, certainly unresolved, and kind of that maybe brings us to like the topic of aging that I would definitely like to discuss. It's a super popular topic lately, both kind of in the popular media as well as kind of the scientific research with all of the headlines surrounding longevity and things like that. So just to begin, do you, in your opinion, or maybe your scientifically valid opinion, but is aging inevitable? And then what do you think might be some of the causes of aging? And we can tie in obviously some of this to NAD, but I know that there are several quote unquote theories of aging, maybe like 5 or 6 that involve many different mechanisms. But is there a current consensus on sort of what causes aging and whether or not it is inevitable? I mean, clearly biologically, yes, it is. We all age and time moves forward, but what are some of the main reasons in your opinion that are accepted maybe as the causes of aging?
Okay, well, first, I think that aging is a fundamental part of animal life, and that I'm not sure whether so-called immortal jellyfish are truly immortal, but even jellyfish would be sensitive to some amount of mechanical disruption and some amount of radiation disruption that would prevent them from repairing themselves. And what I like to think about in terms of animal life is that animals do what we do because we have a gene set that prescribes our shape and our physical properties. So we're not, like, human beings are not capable of sustaining life at 50 degrees C. And neither are most other animals. But if you find an animal that can live at 44 degrees C, you might be able to select for mutants that can grow at 45 degrees C. And if there are genes in that gene set that by mutation and selection will allow the critical functions to still work at 45, then you're going to be able to select for mutants that can do that. And over the course of, what is it, 500 million years of animal evolution, I don't believe that longevity has been a directly selected trait. I think that longevity is an emergent trait from things that have been selected. So what I mean by that is that animals need to be able to identify food and acquire food, avoid predation, identify mates, have reproductive success, and then protect their offspring from predation and provide food for their offspring until their offspring can achieve reproductive success and find food for themselves. That's what's selected in animal gene sets, all animals. Now, further, bonus points if the animals can do multiple cycles of reproduction. Now, there's certain male insects that can only, quote, do it once, but most animals can have the ability to find a mate, reproduce, and then either stay with that same mate and reproduce multiple times, or find another mate and reproduce. And of course, and they've propagated their gene sets for their ability to do that. Out of those multiple cycles, the ability to maintain reproductive capacity multiple times and to maintain fitness, because the animal still has to be able to find food, avoid predation, right? You get longevity out of fitness for all of the other things, right? But you don't necessarily have a direct selection for longevity. And so this was pretty well understood by evolutionary biologists through the 1980s. In fact, Michael Rose, who's a Drosophila evolutionary biologist, put it into a monograph on the evolutionary biology of aging that was published, the initial edition, in 1991, excuse me, by Oxford University Press. This has thousands of citations. This is not unknown work. And so the idea that longevity is an emergent trait and not something that was directly selected is something that was pretty well known. And then when Rose continued and did experimental work in this system using flies, he reasoned that if he wanted to get flies that could live longer than the wild type flies that he had in the lab, what he was going to have to do is keep males and females separate to something like 90% of their expected lifespan, right? 90, 95% of their expected lifespan, then mate them, and then continue to apply that selection for them to be old and still capable, right?
Right,
There's
because
never been a selective pressure on primates to do that.
Yeah, because essentially, I mean, from what you're describing and from, you know, as much as I know, is that essentially once an organism is, say, past the point of reproduction, that's sort of where what evolution and selection no longer really cares. It's, you know, just survive it long enough to propagate your genes, and then after that, it's essentially, you know, you don't matter anymore. So
what comes out of Michael Rose's work when he does the fly thing is that, yeah, he can get long-lived flies by doing hundreds of cycles of this old male and old female pairing thing. And the differences in these old Methuselah flies, or these flies that live longer, are not monogenic. So what are longevity genes? They're all the genes that promote fitness. They're all the genes. For a human, it would be all of the genes that allow my circulatory system to work, that allow my heart to function, that allow me to recognize food, that allow me to chew, that allow me to get from one place to another. So all of the genes, it's not the idea that there are monogenic longevity. Now, there are sort of monogenic longevity genes. There are, in growth factor pathways, like there's loss of function mutants in IGF receptor pathway in worms, and there's the Ames dwarf mouse, which is essentially in the same pathway in mice that are long-lived. These are not fit mice. Okay, this is a dwarf mouse that can't compete with other mice. It wouldn't even be able to acquire food. Okay, so this is not an improved, you know, fitness mouse. This is a growth impaired mouse that can live a longer time due to attenuation of a, you know, growth factor pathway. And so it's not like a dominantly acting super mouse, longevity mouse. And there have been some promotional, people in the field that have acted like, oh, longevity is solved. We identified longevity genes all the way back from yeast, that we find those genes in humans, and their longevity genes in yeast, and their longevity genes in animals, and they're going to be longevity genes in humans, and we have activators of these genes, and aging is a solved problem, and you don't have to age. And that stuff is just not evidence-based. The monogenic longevity genes are not dominantly acting due to what's called antagonistic pleiotropy. These mice are not fit. Some of the reports on what's called the Firco knockout mouse being long-lived, that paper basically should have been retracted. The adipose-specific insulin receptor knockout is not long-lived, even though the Ames mouse is long-lived. It's not a healthy mouse. The so-called longevity genes in yeast are not even longevity genes in both models of yeast aging. They actually failed in worms and flies. They're not really longevity genes in humans. They're not activators of those genes. And we don't understand aging as well as we would like to, but aging is a loss of resiliency, and it's a loss of the ability to repair stuff. And you need metabolism to repair everything, and entropic forces are real, and opposing entropy can be done in living things. We do live, here we are. We're not in technical violation of the second law of thermodynamics, because a Brady-Holmer is not a closed system. You take in food, as I explained earlier, the sun is part of your system. The sun is growing plants. You're either eating those plants or you're eating the animals that ate those plants, or both, and you're acquiring energy, and you're using energy to repair stuff, to oppose the entropic factors. And this even allowed young Brady to increase in complexity during your development. You actually went from single-celled to a born human at 10 pounds or something to whatever you weigh now. You got more brain connections. So you opposed entropy, right? You became more complex. That required energy input, and and your brain can still become more complex, but there are other things in your body that you're going to discover as you age that do have a little breakdown problem. And when your vision gets worse, it doesn't really get better. When when the shape of your eyeballs change, it doesn't have a way to get back to where it was, okay? And so the idea of rejuvenation is easy to say and may not be possible to do. When we can also talk about induced pluripotency and stuff like that. But aging is part of life. Aging is easy to accelerate. It's hard to slow. Probably the optimum diet and physical activity, and not smoking, and not, having guns in the house, and becoming vaccinated against emerging infectious diseases and polio and things that used to go around in the olden days. Those are things that will protect your genetically encoded lifespan capacity. I don't know that we can extend it. And actually, I am an optimist. I'm just not somebody that accepts things that were really disproven in yeast already and that don't fully apply to the human condition.
Yeah, there are obviously certainly a lot of things that you had kind of covered there. And, one of the things that I just wanted to revisit was that just intuitively, when you were talking about aging being monogenic, that, we have longevity or lifespan controlled by these few amount of genes, it just doesn't really make that much sense given how complex we are, like as humans. And then I just think these concepts are so interesting of slowing aging versus this whole concept of reversing aging. It makes sense that we could slow it, because if we have these things that are progressing aging more rapidly, like the environmental damage and DNA damage and all these things that are occurring, there are obviously ways to slow those, whether it's through diet or exercise or whatever interventions that we have. But reversing aging or rejuvenation seems like this totally this process that is clearly more complex. But what would that even involve? I know there are several theories as to like why we age, and then you would have to essentially just reverse those. I know there's kind of like the information theory of aging, that there's like this loss of information, and that you essentially need to return us to this previous like state, and that's what reversing aging would mean. Or you mentioned induced pluripotency, potentially using stem cells and things like that. What does, from a broader perspective, like what would reversing aging even mean if we could do it?
Well, you'd have to have a functional test, right?
Sure.
So, there are people that are trying to develop research tools to try to, monitor aging, right? And so there's a lot to unpack in what you just said. So in terms of, let's say a human being who is aging poorly, let's say that they drink and smoke a lot, right? They have a life trajectory that doesn't look good, or they have an unhealthy diet, atherosclerosis, smoking, et cetera, et cetera. So they could be aging poorly, potentially kind of turn their life around, and they could potentially add years to life and add life to years, to sound like a Hallmark card, or whoever said that. I don't know who said that. But, so that you can sort of improve your trajectory, right? Now, then there's monitoring of biological aging, of which methylation clocks are among the, more active research areas, where people are taking blood samples and they're matching DNA methylation biomarkers with age and age trajectories. And there are people selling kits now, and they saying, "Oh, I reversed my biological age." Unless that stuff is, backed up by a functional test of what you can do now that you couldn't do before, I don't know what it means. And basically, performance is what matters. So if you can run a mile faster, or you can, do something better, then you're doing better. I don't really see the value proposition to a consumer for the age tests, because they're just basically research tools.
Yeah, for sure. And it definitely makes sense what you just said about performance kind of being the main metric of interest. So for instance, if I'm, when I'm eventually, say I turn 40 years old, if my methylation clock tells me that I'm 26, but I can't run a 5K as fast as I could when I was 26, then what does that clock even matter? I'm older because I can't run as fast as I could when I was 26 versus 40, So clearly I've aged in some aspect.
Yeah, clearly you've aged. You'll probably see that wound healing is one of the things that's striking. Strikingly declines, in aging. I don't see people like volunteering to. I mean, there's some people you can do research study and you can pay volunteers to, give them a defined capsaicin burn or something like that, or even a mild heat burn with like a temperature block, almost like a mini branding iron or something like that. And you could photographically measure the healing time as a function of age, and you would be able to show that people kind of lose their repair capacity. In fact, that's one of the things that we're really interested in doing clinical tests of NR to see whether NR will allow people to maintain their skin repair, despite the fact that they are older than they used to be. In terms of some of the things that get talked about the most that are kind of regenerative potential, people are talking about induced pluripotency or, partial reprogramming. So this is where you take, remember when Dolly was cloned, it's sort of before your time, but they took, somatic cell from this sheep, and they, and then Yamanaka was able to show 4 transcription factors. They get abbreviated as OSKM. 4 transcription factors added to somatic cells will induce pluripotency. So take something back to a stem cell that can then differentiate in a cocktail of factors into various different cell types. So you're knocking the cell back. But the thing is, these Yamanaka factors are known oncogenes, okay, which means that they get turned off in development because staying on is associated with proliferation and lack of apoptosis, right? And when you turn them on in inducing pluripotency, you can generate malignant, clones. You can generate teratomas, which are basically horrors that are grown in the laboratory. And when people are doing IPS induced pluripotent work in laboratories, you don't have one cell, Brady. You have a bunch of cells that are bathed in they're CRISPRed in the Yamanaka factors or viral vectors in the Yamanaka factors, or however you're re-expressing them. And you're getting, thousands of, I don't know how many cells under a microscope, and somebody's actually looking at the cell behavior. And if, something starts forming, colonies on soft agar, basically, tumorigenesis on a dish, you get rid of it. If something is starting to form a teratoma, you get rid of it. Okay, so that's very different than inducing pluripotency in a tissue of an animal or in a whole mouse. And there are now reports of 3 Yamanaka factors that were introduced into a tissue where someone claimed it was rejuvenating. But the weird thing is that they declared that they introduced 3 Yamanaka factors into an eye of a mouse while they were writing a book in 2016 to 2018. They named the graduate student that was doing this work. And they said that they intuited that by leaving out one of the Yamanaka factors, that they didn't think that they were going to generate any malignancies. And then lo and behold, 2 to 4 years later, this group published a study that said, hey, no tumors. And then you look in the fine print of the paper and it says the investigators were not blinded to treatment, which means that the investigator whose name was cited in a widely read book as having rejuvenated a mouse without generating any tumors is working for 2 to 4 years in the laboratory, where his or her boss has already told people that what the result is going to be. And can you imagine the pressure on that person if they find some mice that, oh yeah, they did form tumors, or they didn't actually get their vision back? So that doesn't really fit with what the boss said. So it's basically an experiment that was amazing that it was actually peer-reviewed and published. And so I don't consider the non-blinded experiment to be very convincing evidence that 3 out of 4 Yamanaka factors are not teratogenic or oncogenic. And then just in the last week, there was a study where all 4 Yamanaka factors were induced at 2 different points in the life history of mice, one like middle age and one sort of older, for not a very long time. But if you look at the paper, the number of mice was extremely few, like N of 3 and 4 in one of the figures. And so there's no way that you can really evaluate the safety. They didn't say whether or not the investigators were blinded. They didn't really show anything that there's just no way of knowing whether that's really safe or that's really rejuvenating. And if you are rejuvenating a brain and you're epigenetically resetting the brain, and we know that epigenetic marks are part of memories, do you distort what the mouse or human has learned in its 2 years of life? I mean, so it's a research area, let's put it that way, but it's not been shown to be safe or effective.
No, certainly. And, all of what you just said kind of just goes to show that this is a very, very complex research field. There's sort of a lot of controversies, and nothing is really as straightforward as sometimes we would like it to be, or maybe people studying these things would like to make it out to be. And that kind of brings us to the last little topic that I'd like to talk about today. So you clearly, are skeptical of, a lot of research going on in this area, rightfully so. And you've been very outspoken over I would say maybe the past few weeks on Twitter, posting kind of multiple Twitter threads in sort of this debate, maybe we'll call it, regarding, and, we'll just put the name out there, but, a well-known scientist, Dr. David Sinclair, who's kind of very, very outspoken in the longevity field, has gained a lot of popularity, and done a lot of, research. He's a very, well-trained scientist. So can you just give us a brief intro kind of into what you've been so outspoken about on this topic? It relates to, the quote longevity genes that we briefly brought up. It does include a little bit of NAD, which we've extensively talked about. But can you just kind of give us a brief overview of what some of your issues are with maybe not only Dr. Sinclair's work, but maybe some of the claims that he is making about his work, and then we can kind of build upon it from there.
Well, people are interested in longevity. People are interested in lifespan. In fact, Lifespan is the name of a best-selling book by Dr. David Sinclair that has been incredibly influential. I mean, I would say that David Sinclair is the most influential person in the molecular biology of aging.
I would say that's accurate,
right?
For sure.
And globally. And he's been a scientific leader, and he's now basically introduced people around the world to the ideas of molecular aging. So if you look at the front cover of the book, the subtitle is "Why We Age and Why We Don't Have to." And unfortunately, Dr. Sinclair doesn't know why we age and has suggested that we don't have to, in a way that is actually counterfactual. And the whole premise of the book is counterfactual. If you look at the back cover of the book, it says, what if aging is a disease, which is a counterfactual question, and that disease is treatable, which is also a counterfactual question. So the whole thing is based upon a premise of, what if you didn't have to age? What if you could live forever? Wouldn't you want to? So he's using basically a literary technique that was described by Samuel Taylor Coleridge of temporary suspension of disbelief, that he's taking people on a journey in an imaginary world in which we understood aging, and we had monogenic longevity genes that we can activate such that we don't have to age, and aging is now optional because we understand aging so well. Well, if that were the world, then you might consider that aging is treatable. And if aging is treatable, then aging must have been a disease. And so he's got people all, mixed up into thinking that he understands why we age and why we don't have to. The problem is the underlying evidence in the book is all based upon stuff that doesn't work. So, the fundamental insight made by Dr. Sinclair in 1997 was how a yeast mother cell can divide so many times in one model of yeast aging called replicative lifespan. And this is a model in which calorie restriction, growing the cells on lower glucose, gives that mother the ability to divide more times to produce daughter cells more times. The other model of yeast aging, loss of SIR2 gives a longer lifespan. As I've explained on Twitter, in the model of aging that Dr. Sinclair and Guarenti were working on in 1997, the advantage is only to the old mother cell, a cell so rare, there's only 1/2 to the 21st power of those cells in the culture. In other words, there's no survival advantage of the yeast culture to even maintain the function of that SIR2 gene. And loss of that SIR2 gene gives cells that have a longer lifespan in the other model, literally the model in which the cells stay alive for weeks or months. Loss of SIR2 has an advantage. SIR2 is not even a longevity gene in both models of yeast aging. And then David, unfortunately, he and I are on a first name basis. We know each other very well. I'm actually in his book for my discovery of the nicotinamide riboside kinase pathway. At one time, I consulted for his company, so I know a lot about the resveratrol story as well. David is always saying that SIR2 was then found to be a longevity gene in worms. It was found to be a longevity gene in flies, and then it's a longevity gene in mice and in people. And the problem is it was reported to be a longevity gene in worms and flies. And then it took 10 years for that literature to be partially cleaned up by Linda Partridge and investigators from 9 institutions that showed no, it was a strain effect. It's only in particular backgrounds of worms and flies that extra copies of SIR2 are lifespan extending. When you outbreed and you do that in fresh worms and flies, there's no survival advantage. As I said before, there's no reason that you would expect a gene that is important in 1/2 to the 21st power of yeast cells to anticipate the role of thousands of genes in animal longevity, when longevity is an emergent property of animals and is not even a directly selected thing. So the whole idea doesn't make sense from the evolutionary biology of aging. It's inconsistent with the recessive nature of growth factor longevity genes, which are not pro-fitness genes. The resveratrol is not a SIRT1 activator. The things that he still claims are SIRT1 activators, actually still claims that resveratrol is a SIRT1 activator. Those don't help people. They were extensively tested by GSK after GSK bought out Sirtris and all of the technology that was developed by Sinclair. And he basically acts like, the longevity genes were discovered, they were validated in animals, they exist in humans, they can be activated. He claims to have, rejuvenated vision in mice. As I said, the experiment wasn't blinded. It was pre-announced, which makes it, not really believable. And the problem is that rather than, when I bring up these critiques, which are shared broadly in the scientific community, Dr. Sinclair has not really responded. And most recently, he claimed that misinformation about resveratrol was being propagated by myself. And he responded by saying, oh, this will clear it up with a 2006 paper, which is, 16 years behind the times of refutation from other people. So at this point, he's blocked, Dr. Jeffrey Flyer, who's the former dean of Harvard Medical School. He's blocked me. He's blocked Dr. Brad Stanfield, who is a, person that, has a YouTube channel who talks about longevity and the evidentiary basis for scientific claims. And he has an audience from the book and from Instagram and from a podcast that I'm concerned is spreading a lot of misinformation. And so I think it's a real problem.
Yeah. And I mean, there were, I've been aware of kind of all of the recent blocking and things like that. And I remember seeing a tweet from even the former dean of Harvard Medical School, who had quoted Dr. David Sinclair's tweet about, oh, hopefully this will clear it up, where he cited his 2006 paper. And the dean even said, it's very troublesome that you're citing a paper from 2006 that hasn't been replicated or has even been maybe disproven by some more literature in recent years. I mean, that's a paper from 14, 15, 16 years ago. And so, yeah, it seems like it's, a sort of attempt at salvaging, this whole sirtuin as a longevity gene story. And the resveratrol thing as well, it's, as you said before, this whole lifespan book and kind of a lot of the claims he's making is all based on this foundation that we have to accept sirtuin as a longevity gene, and that if we can activate these sirtuins through whatever mechanism, whether it's caloric restriction, fasting, resveratrol, that we can extend lifespan. And sort of if that's not true, then this whole sort of, it's all a kind of facade and this whole sort of foundation breaks down. I mean, is that correct?
It's the book is falsely premised is the problem. And like I say, I don't think that aging is particularly well understood. I think it is a part of life. I think the dominantly acting longevity genes is a myth. The sirtuin activation story is not true. And that even and then importantly, the idea that sirtuins are the primary mediators of NAD is also not helpful. There's hundreds of NAD dependent processes. There's even dozens of NAD consuming genes. And among the NAD consuming genes, like the PARP1, PARP2, and the 5 PARPs that we found that mediate the interferon response in coronavirus infection, there's enzymes called SARM1 and CD38. All of those gene products are more highly regulated than sirtuins. Excuse me. So I don't think that, I think that sirtuins are, moderately interesting. There's been tens of billions of dollars invested in looking at them. We know a bunch of things about metabolic regulation that because of that work on sirtuins, but there's way too much. The street lamp has been lit very, very brightly around CERT 1 through 7. Initially, and actually, I don't have time right now. I have a meeting in 4 minutes. So I might want to repeat this little part and kind of like see where you are after you edit some things and see if you want to come back and finish things. But
yeah, sure thing. Yeah, no problem. Sorry, we've been running a bit over. But I do think kind of we covered a lot of, in terms of that sort of thing, so I appreciate you, discussing that. Okay. So I guess in like maybe the last minute or so, can you just tell people where they can find you if they want to learn more about your work, Twitter, social media, things like that?
Sure. Well, I answer questions most days on Twitter at Charles M. Brenner. And you can also read all of our papers on the lab website, which is brennerlab.net. And I'm going to be doing this research for a long time. I've got a great group of people working with me. And whenever we publish papers, we'll put them out on Twitter. And whenever people publish other papers in the field, feel free to ask me about them.
Awesome. Well, again, thanks so much, Dr. Brenner. This was a great conversation. And again, I appreciate your kind of activism on Twitter, as well as your rigorous scientific work that's going on. So I encourage everyone to give you a follow on Twitter and keep up with this ongoing action on social media and in this longevity space. So again, thanks so much for sitting down with me today. And I look forward to releasing this one.
Okay, my pleasure.
Right.
All right.
Thanks.
Take care, Brady.
Bye. Thanks, you too. Bye.